May I have your attention, please? We'd like to go on with the luncheon speaker, and thank you very much. It is indeed my very distinct privilege and high honor to present to you one of our distinguished professors at the Baylor College of Medicine, who is also a Howard Hughes Medical Institute investigator, a member of the National Academy of Sciences and the Institute of Medicine. She's received the Javits NINDS Award from the NIH. She also is the holder of the Soriano Award of the American Neurological Society. In addition to all of that, she is a consummate physician, scientist, and educator. Dr. Huda Zoghbi will speak to you today about cooperation and collaboration in research. And I think it couldn't be a more fitting topic, especially after the morning program. Dr. Huda Zoghbi.
Thank you, Dr. Alford. It's truly an honor to be standing here today to share with you some of my thoughts about collaboration. I have to admit I'm a little bit petrified to tell you this story from the perspective of biology, but hopefully the principles are the same. I think the guiding principles of collaborations and creativity in science are the same, whether it's biology or physics or space research. So what I'd like to do is perhaps tell you a little bit why I believe collaborations are critical for advancing science, and maybe share with you stories how collaborations have shaped my career and life and made a scientific career for me through fun experience. At least from the perspective of a biologist, a key factors for success in research are first mentorship. That is, for one to start a scientific career, they have to be properly mentored. And the first credit I would give is to my mentors who really prepared me to be a scientist. Of course, it's my responsibility to pursue scholarship and to continue to educate myself in the process. And all of us know it's really the creativity that makes for great advance in science and the best of ideas. Environment is critical, and I've been fortunate to at least grow up as a scientist in a wonderful environment of the Texas Medical Center, and that's key. As an academician, I've enjoyed scientific freedom, and that's something I will never trade, and that's what really keeps me at an academic institution, is that I know if one day I decided to pursue new ideas, I could do that. Nobody will challenge me that this is not fitting with the program. And that's something I always share with young faculty. But perhaps the one I enjoyed and I try to share with my colleagues and young students is the value of productive collaborations. And this is what I'm going to spend some time to tell you about. And of course, we all know that we need money to be successful. And that's the beauty of collaboration, is your dollar will go much farther if you collaborate with someone. And I'll share some examples of that. And finally, I think a lot of things happen by chance, and serendipity plays a role. It certainly played a role in my science as well. There is a big discovery I made was in part serendipitous. I will not be talking about that today, but I acknowledge it's not all calculated and planned. So, as you've heard, I'm a physician, and I started my career as a child neurologist. And as a child neurologist, you see both adult and children with terrible neurological problems. You see people who used to be healthy and then lose the ability to perform simple functions, speaking clearly, walking independently, eating without choking, losing their ability to think. It's really critical. It's who we are. Our brain, the healthy brain, is important. And this was the state of neurology back in 1985 when I finished my training, and I found that to be terribly depressing. So I took a detour after finishing my clinical training and decided to get trained into the scientific method by joining a molecular lab, the lab of Arthur Baudet, to really immerse myself in research, take all the graduate courses, and be trained so that I can help my patients. And I had a very simple idea. Back then, genetics was just beginning to use the tools of molecular gene mapping to try to link diseases to sites on the chromosome so that we can find the genes. So I thought if I go to the lab, learn how to do science, I can map the genes for the diseases I'm encountering, and maybe if I map the gene, I create animal models, study the mechanism, and one day come up with a therapeutic. And I'm fortunate that I've been in every part of this circle, and we've actually now gotten to the point where we've done what we call preclinical trials. That means therapeutic trials in animal models, and we are on the verge of beginning some human patient trials. It took 20 years, but we're there, and I think the technology is better now for us to get at it. So I'm going to share with you two stories that have happened that I really experienced the joys of collaboration. The first one pertains to the first disease for which we identified the gene. It's a disease none of you have heard about. It has a big name, spinocerebellar ataxia. It's called type 1 because it was the very first ataxia genetically mapped. And what ataxia means in Latin is lack of order. So these patients are very uncoordinated. They're healthy for about 30 or 40 years of age, then they lose their coordination, they need walkers, and eventually they deteriorate where they need a wheelchair. They can't speak, they can't swallow in a coordinated way, so they choke and they die. So it's a lethal disorder. And 50 miles north of Houston, Texas, there's this large family, I hope you can see this, that there are multiple generations of affected individuals in this family. And every filled-in circle or square, circles females, squares being males, is an affected person. And anyone with a slash, that person has died. So you would see that this is a lethal disorder. You'll see that it's a dominantly inherited, means if a person is affected, each of their children has a 50-50 chance of having this disease. So I set out to identify the gene of this disease. And one of the things we do as physicians, especially neurologists, take a good history, ask the patient questions, ask about the family, learn something about that. And now you'll see a color-coded pedigree where you'll see that the disease started at different ages in different people. In the older generations, it started in their 70s and they lived into their 80s and 90s. That's pretty good. That's really not disrupting life very much. But as we move from one generation to generation, this disease started earlier and earlier, up to the last generation where I started seeing children who got the symptoms at 4 instead of 60 and died at 10 instead of 70. So clearly, you've got a genetic disorder. There's a gene that's being passed on, but somehow this gene is changing. The mutation is dynamic. It's not static. It's not the same mutation being passed. And that's where serendipity played a role. I was in a conference hearing Tom Kasky describing the cloning of the myotonic dystrophy gene, and the gene at the time was discovered to be mutated through a dynamic mutation, and I'll tell you now what a dynamic mutation. So after having been trying to clone this gene and getting down to 1 million base pairs, remember this is now late 80s, early 90s, pre-genome sequencing area. When you have 1 million base pairs, you have to work your way through them almost a few base pairs at a time. I ran back to the lab and called my collaborator, Harry Orr, in the University of Minnesota. He and I were collaborating because he had more families than I did. I had this one large family, but he had some additional. And through us, we defined that 1 million base pair region. And at that time, any group trying to clone a diseased gene, when they got to 1 million base pair, they wanted it for themselves. Harry and I chose to really continue to work together. So I called Harry, and he said, Harry, I think we should stop looking for all sorts of mutations. We should only look for one type of a mutation, a dynamic mutation. The region is 1 million base pairs. Let's split it in half, but let's keep 70 kilobases of overlap so that we don't miss it. So Harry and I agreed that we had a strategy to do that. And we did. And on April 8th, 1993, we identified the mutations in both of our labs on the same day. And the mutation happened to be in the 70 kilobases of overlap. So Harry always likes me to tell people is that discovery and collaboration is what got us on the top fold of the Wall Street Journal. We made it to the top fold, not because this is an important disease, it's a very rare disease, but really because of the style of the collaboration. True openness, true sharing, not leaving any chance that one has an edge over the other. That was important. So this is just a schematic of the gene. And you'll see that the part of the gene that makes the protein is colored in yellow, and in that there's a little pink triangle. In that triangle, there are 3 bases of DNA, CAG. These bases of DNA encode for an amino acid called glutamine. And anyone in this room may have 25, 30, 35 glutamines. That's normal variation. But in these patients, the glutamine goes beyond 35 to 39. And up to 82. And now if we look at our patients, we understand this dynamic mutation. You'll see that in normals you can have 10, 20, or 30, but in patients where there are 40 repeats, you get disease onset at 70, and in those with 50, you get onset in the 30s, and finally the children had 70 repeats. So this repeat, which expanded as the basis of the mutation, basically, as it moves from one generation to generation during the replication of the DNA, it gets bigger. And now the dynamic mutation explained to us why the age of onset kept getting younger and younger. So that was really opened up the field, and there are now 9 different disorders that are caused by a similar mechanism. Huntington disease was discovered the same year, was also caused by the same mechanism. And just to show you a picture of Harry, this is the year we discovered the gene. We were both much younger, and our collaboration is still ongoing since 1988. So I make the point of saying that because both of us are very committed individuals. We both run labs. We both train people. We work on an extremely rare disease. And yet we manage, by both of us focusing on that disease, to really find ways to carve niches for our students, for our fellows, and for ourselves so that we can continue to get funding. He has an NIH grant on his work. I have one on my work. And if we could do it for SCA1, I think anyone can do it for space medicine, for space research, for anything, because really these are vast fields and they need such collaborations. And we'll talk maybe briefly about what are the key factor that has played well in us navigating a successful collaboration. Just to tell you very briefly what we wanted to know. So I told you if you have 35 glutamines, the neurons are healthy. And if you got 40, very few, imagine a poem with few extra words. It's going to ruin the poem, and that's really what's happening. This protein is there, but just has a few extra glutamines, and that's causing massive degeneration. So we've been working on that, and the way we've done it in this collaboration, we've made sure we always divided the labor. We try not to replicate the same experiments. So Harry and I, whenever it's time to renew our NIH grants, I call him up and I say, these are the aims I propose to do for the next 5 years, because I know what he's doing currently. So we really coordinated long-term vision, and we also coordinate on a month-to-month experiments. We also are very sensitive about authorship and acknowledgments, in the sense that a student in my lab is going to create a mouse model. That student deserves to be the first author. Harry can use the mouse. His students can use it and do other things with it. But we coordinate who is the first author on which papers. And fortunately, our students always got their pick of first authorship based on the ideas they pursued with these projects. And that's, of course, important for career advancement. Students want pieces of the project to take with them, or fellows. We've always worried about that. And that has helped us continue the funding and hopefully continue the successful careers of trainees. The collaboration that Harry and I had and enjoyed, quickly it became clear that we need to branch out of the realm of biology. And I think this is what's happening in biomedical research now. We're starting to need really people like you. We need the mathematicians, we need the physicists, and quantitative computational biologists. So we started analyzing our animal model and realizing we have millions of gene expression changes in these neurons. We have various protein-protein interactions. We have various types of protein DNA interactions. And when somebody like me is hit by a million pieces of data, I'm used to very simple biological linear pieces of information. That's when we reached out, and we've been fortunate to collaborate with Laszlo Barabasi, and then with two younger quantitative biologists and mathematicians, Chad Cho and Wei Li. And just show you one example here. This is a network of proteins, and the proteins in blue, the dots in blue, are all the proteins that cause a similar disease to the disease I told you about. There are over 50 different inherited balance disorders or ataxias. And in my lab, Jung-Hoo Lim identified the partners of these proteins. And when we connected all those together, we found they all actually interact with the same group of proteins. And it's not surprising when you think about you and your best friends, you probably share a lot of people in common, and it makes sense that proteins that work together cause similar syndromes when they are mutated. But here we really had to recruit the help of mathematicians and physicists to help us with these data. And what came out from these studies, I'll just give you one last biological example from the ataxia study, is we learned that our protein, which is the big orange thing, when it's normal, it has many interactors. But here are two examples of interactions where the blue one does not require phosphorylation of the protein, but the green interaction required this little phosphorylation group to sit and the protein be phosphorylated. And what we've learned that in the brain, it's a balance. The protein spends part of this time with the blue partner and part of this time with the RNA binding partner. And what happens in the disease, just having those few extra glutamines makes it look as if it's always phosphorylated. And what happens is that you have a lot more of the protein spending its time doing those functions over the functions mediated by the blue-orange interactions. And that's really important because that defined for us a phosphorylation site we can go after, inhibit that phosphorylation for therapeutics, and screen for things that prevent such excessive interactions. And finally, just to mention, of course, the reward of such a collaboration. So it took us, Harry and I, about 20 years to get to that, or 15 years to get to that point. But the reward is that I think we've accomplished for this one disease a lot more than maybe 100 labs have accomplished for some other neurodegenerative diseases, although we're both relatively small effort on this particular area. One thing I'm passionate about is maximizing use of resources. I think NIH money is taxpayer money. It should be put to good use. We should not be doing Me Too type of experiments than investigators using NIH money. We should really leverage the resources so that everybody does the best possible experiment and collaborate. It's fun. Harry and I have team spirits. When we run into each other at meetings, we're the two big groups working in this rare disease. We enjoy seeing each other. We look forward to it. We make dates. He came to Lebanon to visit Lebanon with me. And it is, it's a way for us to brainstorm, to be creative together when we meet at meetings. I always tell people, find who's working in your field, try to collaborate with them. It just makes it so much more fun. And there are financial rewards to collaboration. I did mention another collaborator, Juan Botas, but through our collaboration and the ideas and progress we did, we were able to compete for a very nice and large award from the Howard Hughes Medical Institute called the Collaborative Innovator Award, which my partners, Harry Orr, Juan Botas, and now Trey Westbrook, a young creative scientist, joined us. We're now doing large screens to find drugs that may inhibit that phosphorylation and factors that can modulate it. So this is one story that was totally enhanced by a collaboration. And once you do this and you discover this reward, you start really using this in your life much more frequently. And I'm going to tell you now about another collaboration. Some collaboration can be intellectual. They don't always have to be day-to-day lab. And reaching out for people a little bit outside your field is really where great ideas come from. And this other story I'm going to tell you, and I'm borrowing here on Théophile Gautier, L'Art pour l'Art, Science for Science Sake, in that it was really born out of just interesting, can I do science for science sake, not applied to disease? I'm a neurologist. I studied the ataxia, I studied another autism disorder. I wanted to really see if I can just let my curiosity run with it. But I wanted to be sure I work on something important and interesting. So I went to Hugo Bellin, a professor at Baylor College of Medicine, who's a Drosophila biologist. And I, of course, was dealing with mice and humans. So I told him, Hugo, do you know of a great gene in Drosophila that's important for the nervous development of Drosophila? Just it does something important for the fly that the fly can't live without. And maybe if there is such a gene, maybe I can find its homologue in mice and humans, study it and see, does it do similar things in the brain of mice and humans? So Hugo said, yes, this is Hugo Bellin. There is a very important gene, and this gene was cloned in a lab in San Francisco, Yunnan Jan's lab. And what he discovered, this gene was called atonal, because without it, the fly is deaf and uncoordinated. And you'll see all these areas that are in yellow. These are areas where these mechanoreceptors or chordotonal organs that allow the fly to know its place in space, where its wings are, where its legs are, and it can also hear through that organ on its head. That's called the Johnston organ. So this is a gene that makes up all the neurons in this yellow spot that's very important for hearing. So back in the early 90s, finding a fly gene and going after it in mammals was not quite popular, and nobody thought it would work. This is where I was fortunate to have the Howard Hughes funding, where I could take a risk, and if it failed, they were not going to penalize me as much. So I went for it and found the mouse gene and studied where the mouse gene is expressed. And you'll see this blue area. We can mark where the cells, where this gene is expressed. It's the developing mouse, and you'll see it at the very back part of the brain and the top of the spinal cord. Very exquisite areas. We also found a tiny spot in the inner ear. Very tiny speck of blue in the inner ear. I knew nothing about inner ears. My specialty was the brain. But with the help of faculty in Dr. Alford's department, pursued what is that in the inner ear? What is this gene doing in the inner ear? Well, it turned out to be the gene that makes the cochlear hair cells and the vestibular hair cells that are very important in the inner ear for you to be able to hear this conversation and, of course, to balance, know the position of your head through the vestibular system. And if you look on the left, this is a scanning electron microscope that shows these bundles of inner ear hair cells that are totally lacking in a mouse that does not have this gene. We engineered a mouse that doesn't have this gene. We had no clue when we started with this gene that it's going to be the gene that actually make those inner ear hair cells. And we showed that this is necessary. Without this gene, you can't make them. And subsequent studies in rodents showed it's sufficient, which means if you take a guinea pig and you damage their inner ear hair cells, they'll become deaf. If you, through gene therapy, put this gene just in the inner ear, in the cochlea of the guinea pig, you make new hair cells, and they are functional. The animal can hear. So of all of my discovery, the first one that's going to go probably into humans in translation is this discovery, because it's now been taken by Novartis, that's putting a large sum of money to do clinical trials in non-human primates in preparation to go to human. As you know, there are 20 million Americans who are deaf because of damage to these subtle, tender hair cells, and this is really a great way to do that. In addition to this going back to the back part of the brain, we were able to trace the cells. This is the back part of the brain has a lot of neurons that divide, and then populate our brainstem and the cerebellum, the balance organ, as well as the brainstem where the breathing centers and many key centers are. And by tracing the cells that come from that part and that depend on this gene, we found that the 38 different little areas in the brainstem that depend on this gene. And what's really beautiful, they all serve this one integrated function. And here's the function. We talked about the hearing and the vestibular system. Of course, once the sound comes from your inner ear, it has to go to the brainstem. And those neurons also are specified by these genes. But it's also very critical for the proprioceptive system. And that's the way you know your position in space. If each of you was to close your eyes this minute immediately, you'll still know which way your head is tilted. You will know where's your left leg. You'll know where's your right arm. You don't have to look. That's your inner sense. That's your sixth sense about your body parts. That's proprioception. It's also critical for interoception, which is knowing what your internal organs are doing. This gene makes the neurons critical for that, as well as neurons critical for breathing and arousal. So, of course, we've made mice that don't have this gene. These mice are obviously, they're dead eventually, but when we take it only from one part of the brain, we know that without it, one cannot hear, one cannot localize where the sound is coming from, one has no spatial orientation and balance, and one will not be able to know where any of their body part, will be constantly dizzy, and eventually will not be able to feel a full bladder. In the middle of the night, when one feels a full bladder, it's the neurons made by this gene. But not only that, when you feel a full bladder, you have to get up. So the neurons that wake you up also are dependent on this gene. So it is truly, and of course for breathing, it's critical for breathing. It's a master regulator. And the beauty of it, and we've done this experiment taking the fly gene, putting it in the mouse and vice versa, evolutionarily conserved from the fruit fly all the way to mice and humans, the same system. What the fly needs to do, this one gene, to hear and know where its body parts are, we have the same gene. It does all the same thing, including additional sophisticated senses, such as interoception. So these are two stories that I wouldn't have been able to do successfully without my collaborators. Hugo was great picking the perfect gene for us to study. He made the great suggestion, and for that I'm grateful. Just a couple of thoughts about, people ask me, what really makes a collaboration successful? Trust. I think you have to trust the person you're collaborating with, and with that, you have to be very honest. When I call up Harry and I tell him I have a great idea, I don't hold back. I say the whole idea. Because if I don't and I hold back, one day he's going to get the same idea and maybe give it to a student, and that's where conflict would arise. By me telling him what my idea is, what we would like to do in the lab, I share everything. He knows the boundaries, and therefore he will have complementary ideas to be done by his students. And this is where most people really, where things fail. People might collaborate, but they'll share 75%. It's the 25% that will give you the greatest headache if you have not shared it from the beginning. So I think you have to trust who you're working with, you have to know them well, but then do it all the way. I believe in generosity. I think it's always, tempting to hold on more than you can do because you're afraid, well, what if this doesn't work? Just be generous, let things move forward, share information, things will advance, there'll be plenty for everybody to do. And that's, again, a really important quality. And if you propagate a culture of generosity in the lab, then people in the lab will share and your own work will advance faster. That's important. Also, of course, fairness, making sure you acknowledge if somebody's taking something, you don't infringe upon that credit. And chemistry. I don't think you can just randomly decide to collaborate with everybody at the table you're having lunch with today. It has to click. People have to like to work, know each other, know that they can trust each other. Some personality work better with one type of partner, some with others. So I think chemistry I believe is important. And one thing we forget, that the only way collaboration will be successful is if institutions will reward them, if department chairs, bosses in a company, in any institution, because at the end of the day, when you collaborate, often the credit really go to two people, and these two people have to be individually promoted. And if somebody says, well, you didn't do anything on your own, it's going to be very hard for someone to buy into collaborating. But if people will reward people who've collaborated, I think it's really much easier to propagate that culture and encourage people to be generous and collaborative. And I think the same applies for funding agencies, because if NIH review panels will say, well, this person published 10 papers, but they have Harry Orr with her on all of the papers, then we don't think she can do anything on her own, then I would have never advanced. They have to really acknowledge, and it's up to us investigators to point, we did it together, but here's what I did, here's what he did. And I think it's important to educate review panels to be sensitive to that. And as I mentioned, the chairs, and of course, awards. Often investigators get awards, and it's really important, again, to either do awards jointly or at minimum, make sure both are being recognized for what they've done. So this is really all I wanted to share about collaboration in biology, and towards the end I have a nice story to tell you about the collaboration between space and a biologist. And this is a true story. It's a wonderful interdisciplinary collaboration story between the late Michael DeBakey and Daniel Goldin, who I think most of you know was the longest NASA administrator in the 90s. Michael DeBakey wanted to develop a heart assist pump so that it can help failing hearts until these patients can have transplant or other surgeries. And the type of pump he wanted was a continuous flow turbo pump. My understanding that those don't always work with fluids as well, especially special kind of fluids like blood. What the challenge Michael DeBakey was facing was that the blades were causing the blood to break. The roughness on the blades was causing the blood cells to rip. So there was hemolysis. And the type of currents and the stagnant zones within this pump was causing the blood to just get stuck in certain areas and clot. So he was challenged and being very frustrated, went to Daniel Goldin and asked him, I'm running into these problems, do you have any advice for me? And Dan proposed two solutions for him. He said, the first thing you need to do, I'm going to put you in touch with people who understand finishings of surfaces, because they might give you a better idea on a better surface. And the second thing, it's easy. This is something you may not know, but there's something called computational fluid dynamic analysis, where you basically, in a computer, plug in the properties of the fluid you're trying to run through the pump, and it'll give you some parameters so you can avoid these stagnation zones. So Michael thanked him and went on his merry way to meet those people and do the analysis. And 2 years later, Dan Goldin was at the University of Padua, where he's getting an honorary degree. And the night before, a Contessa threw a party in his honor. And he was at her home enjoying the dinner, and the phone rings at 10 p.m. And the servant comes and says, Mr. Goldin, this phone call is for you. He was a little bit surprised to get a call at her home at 10 p.m. at night. And the person on the other line was Michael DeBakey. And he said, hello, this is Michael DeBakey. Just want to tell you, I just finished washing up after surgery, having implanted the assist pump that you helped me improve upon. And Dan, to this day, will never forget that story. And I think this very simple story really has many of the elements that I was trying to share with you. Intellectual contributions are a collaboration. Reaching out of your own field, you can find simple solutions. If we all get stuck within our own area of expertise, it's very hard for us to advance and push new boundaries. So Michael went to somebody totally out of his field. And thirdly, really remembering to give credit and acknowledging and thanking. For Mike to track Dan in Italy in some stranger's home and make sure to tell him, share with him the thrill of the moment, I thought was incredible. Speaks how we should all do it. So in keeping the DeBakey tradition, I'm proud to say we're trying as hard to do that from our end with a new neurological research institute that I'll be directing at Baylor and Texas Children's Hospital. I'm very proud to say that I'm discovering the joy of working with people at Rice through the research collaborative building. And we actually have 3 faculty, 2 of which already were done with the plans to hire them jointly. These faculty will spend 25% of their time at Rice and 75% in my institute, helping us solve neurological and brain-related circuitry and computational problems, but at the same time, they're in touch with the mathematicians, engineers, and computer scientists at Rice. And this is really the best way to advance science, to have this kind of cross-fertilization. And I'm very proud that we've just hired the first physicist and mathematician jointly and working on some more. Thank you very much for your attention.
We don't really have time. But I did want to say that, excuse me for walking in front of you, but you can see why she's so highly regarded in our institution. And she listened to some of the last panel. David, let's see, are you still here? I'm sure that it opened the door to a lot of opportunities in everybody's mind, having heard your address just now with respect to working together and how we can make great advances through cooperation and collaboration. And thank you very much, Huda. George, I guess you were waiting on your...
Perhaps I'll make a couple of comments about what's going to happen at 1 o'clock. Last year, as you recall, we had a very, very successful two-way link-up with Dr. Christian Otto, who was on Mount Everest. And Christian did an absolutely outstanding job, both in the Everest expedition, but also in demonstrating the use of remote ultrasonography and being able to share that experience with us here during the ISMS. Absolutely amazing. Well, this year, we're not to be outdone. We have another event. This time we have Chris Hadfield, who's a Canadian astronaut. He's a commander on board the Aquarius Undersea Research Habitat, commanding Nemo 14. And Chris will be joining us in a couple of minutes. We're just going to get set up for this. And he'll be live in front of us, able to talk about the value of Aquarius as an undersea research habitat and the type of work that's going on there. So don't leave. If you do get up, go and get your headset, because you might want to have translation available for this if need be. But certainly don't leave, because we'll be getting going in just a couple minutes.
Dave, this is Chris. I hear your voice. How do you hear me?
Yeah, you're right here in this chair. Okay. Here in front of you. We're still going.
Well, Chris, I hear you really well, and I've got you there. We've got you up on the big screen behind me. So despite the fact that you're looking at me, all of us are looking at you. I just gave a very, very brief introduction about Nemo and talked about the very successful mission that you're having on Nemo 14. Congratulations to you and the crew. Great work. And I thought we'd turn it over to you now to talk to our group about the value of Nemo as an analog environment, the type of work you're doing, and how that's going to help us with the future of space exploration.
Great, Dave. And just to double check, we're a little bit ahead of schedule. Would you like to start now? Is folks ready?
Yeah, we're ready right now. I think we'll get going a little bit ahead, and take your time. We've got about 15 minutes or so allocated for this.
Okay, super. Well, first, thank you very much for inviting us in to the conference today. We really appreciate it. There are 6 of us living underneath the ocean. You'll be able to see in this window beside me, as I'm speaking sometimes, as the fish go by. This glass is about 10 centimeters or 4 inches thick, and the wall is 3/4 inch steel, and we're inside a steel bubble 60 feet below the ocean off the coast of Florida, which Dave has probably told you in the intro. There's going to be people walking back and forth through while I'm talking, just because we're getting ready for 2 spacewalks right now. My name's Chris Hadfield, and I'm on this crew with I'm an astronaut with the Canadian Space Agency. I'm on the crew with another astronaut from NASA named Tom Marshburn, 2 engineers that have helped design spacesuits and surface exploration equipment, and 2 technicians that are responsible for the health of the vehicle. So we are 6 people. For 2 weeks, we're staying underneath the ocean for 14 days. In that 14 days, we do 13 spacewalks each, it turns out. So whatever 13 times 4 is, some like 52 spacewalks or something like that. And we're just finishing up now. We've been here for 12 days or so, and we're doing the last of our spacewalks today and tonight. And then tomorrow we will slowly lower the pressure here back to sea level, and we'll get back up into the sunshine on Sunday. Now, let me just tell you why we're here, and then I'll talk about some of the experiments we've been doing. We're always looking for better ways to train for space flight and to prepare ourselves and our equipment for space flight. So this is an analog, much like all of the simulators we use all around the world. I've spent a lot of my life, as Dave has, in simulators in Houston, Texas, both underwater in the pool there, but also the shuttle simulators, space station simulators, robotics, Rendezvous. Also in simulators in Russia with the Russian spaceship, the Soyuz. This simulator here is at the bottom of the ocean because every time we go outside, we can use the buoyancy of the water to simulate weightlessness. And so we can weigh ourselves down to simulate the gravity of Mars, or to simulate the gravity of the Moon, or even to simulate the negligible gravity of an asteroid. And we've been doing all of those things during the 14 days that we're down here. But one of the other interesting findings, maybe more relevant to a medical community, is that the changes to the human body and some of the processes of adaptation we go through when we live and work down here for 2 weeks, we're finding, interestingly enough, to be very similar to the changes that happen to the body during a 2-week space shuttle flight or during the first few weeks of a space station flight. So the exact same experiments that Tracy Caldwell and Soichi Noguchi and the crew up on space station, as well as Ken Ham's crew on Atlantis right now that are docked, the same experiments that they are doing, we're doing down here underwater, partially to train us as astronauts, but primarily to better understand what is caused by the cycle of activities, what is caused by the type of people that we are, the subset of the population, and what is caused by microgravity or the spaceflight experience, having a look at all three. I've collected on the table just some of the things that they're asking us to do in order to gather data medically on us. But first, I just want to show you what happens when you bring something down here without punching a hole in it. This is a can of Pringles that they forgot to punch a hole in, and it's just a very vivid demonstration of the change in atmospheric pressure down here. We're at about the same pressure as in your car tires down here. In fact, it's harder to talk down here. You have to move your tongue and your jaw differently in order to speak clearly down here. You can't whistle. The air is the wrong density to whistle. When you drop a tea bag, it flies and floats because the air is more dense. And so water boils down here at 260 degrees. So there are a lot of fundamental changes to the environment that are also affecting the experiments that we're looking at. Let's see, what do I have? Of course, we're collecting body fluids. We do blood draws prior to the event, during the event a few times, and afterwards. And it's fairly safe. We do saliva samples as well, blood draws, both prior, during, and after. We're also swallowing transmitting pills, same sort of ones that we've used in when we do the high temperature experiments in the Soyuz out in the Black Sea. But we swallow these transmitting pills, and then there's a sensor that keeps track of our core temperature as these pass through the system. They're looking at cardiac output or cardiac rhythms overnight. It's quite an interesting one in that they've built a special shirt that has this running down your chest, and it's got 3 leads. And the way the shirt is built and the way the clip-in shirt is built, you just put on the shirt, Velcro in the 2 collectors. The leads naturally go to the right place. It's hard to misplace them because we're not all doctors and we're not all nurses, so we wouldn't know for sure exactly where things go. But just by the straight design of it, it ends up always being lined up properly, and it plugs into a recording device, and the data gets kept on a chip that will be downloaded after the flight. So we have that. We're also wearing a new experiment here, which I think has a lot of applications, not just in an extreme environment like this one, but in the regular commercial world. It's held in the middle of our chest, by just this simple over-the-shoulder arrangement here, like a backpack, centers it on the chest. It's contained inside this box. It's just a transmitter and a receiver and sensors inside a box, but it's a radar that sends a signal into your chest and looks at the reflections in your chest, and from that can actually measure the blood flow in and out of your heart. Completely non-invasive. And they just did a several-minute calibration of each of us above ground. You can wear it over your shirt. You just put this in place. You turn on a computer that has an antenna. This is sending a radar signal in and out of our chest, and then it's transmitting the results to just a regular notebook kind of computer that has an antenna on it. And then that notebook computer is sending the data back to our medical technicians back topside. So they, on a non-invasive basis, while we're walking around, they are getting real-time, multi-layered kind of data as to our blood flow and our cardiovascular and our lung operation, just purely through this external sensor. So there's a whole suite of stuff going on. What I didn't show you was on these laptops, we also are testing for reaction time. It's exactly the same as the crew did this morning up on station. I was reading in their notes. But they're interested to see first thing in the morning, just prior to a simulated spacewalk, after a simulated spacewalk, looking at how a regular work cycle, a fairly demanding, many days in a row regular work cycle, affects your reaction time, affects your mental attitude. So we're doing reaction time tests, we're doing various psychological surveys, and we're doing it with all 6 crew members who are from all different backgrounds to try and get as big a sample size and as much data as possible, so that when we come out of here after 2 weeks underwater, we've been looking at physical adaptation to compare to space station, how we've dealt with it mentally, how we've dealt with it psychologically, and then outside, we've collected over 3,500 data points on spacesuit design, as well as done 2 full days of exploration spacewalks as if we had landed on the surface of another planet or an asteroid and gone out and collected samples and worked on behalf of geologists back on the surface of Earth. One final thing. The first week, we worked real time as if we were on the moon, so that we could talk back and forth. You just pick up the radio and you could talk back to your support team. The second week, we have worked as if we were as far away as an asteroid or Mars, so we've given ourselves 20-minute delay. So every time we want to communicate, when you send a note, you time tag it, and they can't open it for 20 minutes. Then they deal with it, they send it, we don't open the response for 20 minutes. And so we become a much more autonomous team. And we're interested to find out the psychological and the operational impacts of working in a team that has autonomous control of their own destiny, what it is that we want to do. We can't call back and ask, hey, how do you want us to change this? What do you want us to do in response to a different set of priorities? What do you do when one of these breaks? We've been working just as if we were on another planet. We've got a basic level of skill and training, and then we have to come up with our own conclusions and operations. So that, in fact, also becomes part of the experiment. So it's a real large suite of operations we've been doing. Hopefully, a lot of the conclusions we've been drawing will be helpful, of course, on an exploration side, on a spacesuit design side, but also on a biomedical side, trying to understand and pick out the changes in the human body that occur because of a place like this, and that we can expect to see when we go to other planets and send people out on similar profiles there. So Dave, that's a summary of what we're doing in Aquarius. We're the NEMO 14 crew, NASA's Extreme Environment Mission Operations, a contrived name for Captain Nemo, but it's a good name. And we've been extremely happy with how the 2 weeks have gone. And the guys are just about to go outside for the last daytime spacewalk now. We'll have 2 nighttime spacewalks tonight, which is quite a thrill to be down on the reef, looking at everything that comes alive at night. And then, as I say, we'll start the very slow process of depressurizing, letting our metabolism readapt to to the sea level pressure and then come up Sunday morning.
Excellent. Thanks very much, Chris. Outstanding description of Aquarius as a research platform and all the fantastic work you're doing. Great life science experiments that you have on board. I'll just ask if there's one or two questions in the room, perhaps, if you have a couple of minutes, and we'll see if we can answer some questions. Does anyone from the audience have any questions? Speak up, and then I'll repeat the question so Chris can hear it and answer. Any questions or comments at all? Jim, did you have one? Chris, Jim Voss says to say hi, and we also, I'll ask the question on the behalf of the group, but as we go forward in the next decade of space exploration, the big challenge we face is the economic pressure with the funding for the space program, the funding for analog experiments and things. You've flown in space, you've done spacewalks, you've got a lot of experience, you've now been working on the Aquarius undersea platform. Do you see that there's value for us scientifically in using Aquarius as an analog platform? And if so, is the economic model sustainable to continue using Aquarius?
That's an excellent question. Bang for the buck. What's the right way to spend your money to get the maximum return? All simulators cost money, and you have to question on their product, of course. Is this a simulator that you need to build? How accurate does it need to be? What areas does it need to be accurate in? How much does it cost to maintain it? One of the other ways to simulate spacewalks and evaluate this type of equipment is to use the underwater facility in Houston. But strangely enough, because it's a full indoor facility, it ends up being order of magnitude more expensive than using one that already exists here on the sea floor. It would seem counterintuitive, but actually from a cost point of view, because you don't have to pressurize, depressurize, and repressurize, you don't have to maintain the whole facility to keep the water as an unnatural resource. All you're doing is maintaining the facility for people. This one is actually cheaper than our underwater facility in Houston per man hour when you want to run, and the more you use this, the cheaper it gets down here. The other part is, how do you get a crew into a realistic enough environment that you can draw valid conclusions? We could all just lock ourselves in the room that you guys are in right now, close all the doors, and stay there for 2 weeks. But it doesn't give the same psychological impact. You do not buy into the fact that you can't just go out the door if you really need to. Here in Aquarius, we are physically isolated from the rest of humanity. We cannot go outside and go up to the surface. It would kill us if we did, or at least cripple us with the bends. So that carries its own psychological weight to know that you as a crew of 6 are living in a less safe environment, in a hostile environment, and that all of your actions determine your own future. And so that type of situation is not that easy to build any other way. The Russians do it in vacuum chambers, but that, of course, has a lot of overhead, and you can't sustain it for nearly as long. You can sort of simulate it through expeditionary exercises like outdoor leadership schools and such, but it doesn't give that sense of true limited isolation of contact. We could just as easily be looking out the window and seeing the world, or looking out the window and seeing the gap between here and Mars. The basic environment would be very analogous, very evocative of it. So it's difficult to get this type of environment any other way, and to have one that is built, already exists on the seafloor, and the cost is shared jointly between NOAA, who owns it, the University of North Carolina, the Canadian Space Agency, who contributes regularly to programs here, and NASA, to be able to use a facility that exists, spread the cost over multiple agencies who all get benefit from it. I think it ends up being a very efficient way in order to gather some of the science and the technology and exploration research that needs to be done.
Excellent. Well, thanks very much, Chris. My congratulations to you as commander, to the rest of the crew. You're doing outstanding work. The event that we did 3 days ago in Canada was very well received. The students loved it. And again, congratulations. Keep up the great work. Thanks.
Thanks, Dave. Nice to talk to you. Tell Jim Voss, he's a former crewmate of mine. Jim, nice to talk with you. Hope to see you in Malaysia. Bye-bye.
So you can see the outstanding work that we're able to do on the Aquarius Undersea Research Habitat. The reason why I asked Chris the question that I chose, of course, is in these times we're always evaluating the economic value, the return on the platform that we're getting. Chris was able to articulate the type of research that they're doing on the Nemo 14 crew, but just to comment on a couple of highlights of the Nemo program, which goes back to 2001 when we did the first Nemo mission, now 14 missions over that last 9-year period, a truly remarkable collaboration. On Nemo 7, we saw the world's first remote telerobotic surgery with surgeons located in Canada and the simulated patient underneath the surface of the ocean. When I say remote, I mean in the context that the patient was remote and isolated. Prior to that, there had been demonstrations of telerobotic surgery between continents, but never with that degree of isolation of a simulated patient. Dr. DeBakey came and requested to see what was going on in our Mission Control Center for NEMO that we had at Johnson Space Center. He watched the surgical procedures that were taking place between McMaster University in Canada and the Aquarius Undersea Research Habitat, and at the end of it he said, you know, what I've seen today is among the most remarkable demonstrations of surgical technology that I've seen in my career. And you could tell right away there was a but coming, you know. And he says, but you have to understand that going forward and providing medical care in these environments, you have to be able to deal with the complete patient. You have to provide the preoperative care, the anesthesia. Not only do you deal with the surgery, but the postoperative care, and you have to look at all those elements and integrate them together if we're to be able to perform these types of procedures in these austere environments. Subsequent to that, on Nemo 9, we were again able to test and evaluate telerobotic surgery, this time taking the surgical latencies beyond 1 second, a barrier that people thought was previously impossible to break. The reason why we do this is to do exploration-enabling research that will take humans beyond low Earth orbit to start to answer those questions that Brewster was pushing on earlier. What do we need to do to be able to extend that time-distance constant of human space exploration? Earlier this week, Chris and I did an event where I was sitting in an elementary school in Canada in front of 800 students, and Chris was outside in the water column doing a simulated spacewalk, and he spoke to the students in the classroom. Truly a remarkable experience that captures not only the research element of Aquarius, but the educational value as well. And on Nemo 1, the first undersea research mission that NASA did in collaboration with NOAA, Bill Todd and I were able to talk to 9 million students in America over the internet using this as an educational platform. So my perspective, I'm highly biased. I will admit it right firsthand. It's a valid platform. It's a great analog. It's cost effective, gives us good science, and it gives us strong educational return. And I'm very proud to say that on June 15th, I'll be at the Canadian Embassy in Ottawa, in Washington, presenting to NASA and NOAA a NEMO medallion that was underwater on my Nemo 9 mission that lasted 18 days, but also went to the International Space Station with us on STS-118. We have the NOAA 200th anniversary emblem that was also on board the space station that will be returning to NOAA. And we're doing this to celebrate the effective collaboration between NASA and NOAA to use this remarkable undersea research platform with international collaborative research for the betterment of space exploration. Thanks very, very much, and I think we have a couple of minutes for you to finish the rest of your meal and get ready for our next session. Thanks.